Hello, I'm Sadra Shahdadian, and I'm pleased to present our research on functional and structural neuroplasticity of the somatosensory system in hemiplegic cerebral palsy. In this study, we aim to better understand how the developing brain reorganizes itself after an early neurological injury. Our main focus was to see how these reorganization patterns differ depending on the timing and type of injury, and how these differences eventually influence the clinical outcomes we see in children with hemiplegic CP. The clinical presentation of hemiplegic CP is highly variable, largely because the developing brain has a very high capacity for plasticity. We focus on two major lesion groups. The first was preventricular lesions, which usually occur during the early third trimester and mainly affect white matter development. And the second was cortical subcortical lesions, which occur later in gestation and involve the loss of gray matter. Because these injuries happen at different stages of brain development, we hypothesized that they would lead to different patterns of structural connectivity and functional cortical activation. For this study, we recruited 24 typically developing children and 22 children with hemiplegic CP, including 7 with cortical subcortical lesions and 12 with preventricular lesions. In our clinical assessments, we observed clear differences in sensory and motor performance between these lesion groups. Children with cortical subcortical lesions showed more severe impairments in the function of the parietic hand compared to the preventricular group. To study cortical dynamics with high precision, we used a multimodal imaging approach that combined magnetoencephalography and high-density electroencephalography. This approach allows us to generate high-resolution maps of the brain's response to tactile stimulation and helped us track the rapid spatial and temporal flow of information within the somatosensory cortex. We also quantified the laterality of brain activity using the somatosensory lateralization index. In this measure, +1 indicates completely contralateral activity, and -1 represents completely ipsilateral activity. Our functional results revealed major differences in how the brain processes sensory input from the parietic hand. In the cortical subcortical group, we observed significantly reduced and delayed cortical activity in the primary and secondary somatosensory areas of the affected hemisphere. In addition, the somatosensory lateralization index showed a major disruption in the normal interhemispheric balance in cortical subcortical patients. In contrast, the preventricular group showed activation patterns and lateralization scores that were much closer to those of typically developing controls, suggesting better functional preservation despite the early injury. To investigate the structural pathways supporting these functional findings, we used tractography and isolated the ascending sensory fibers and the sensory commissural fibers. The tractography results showed substantial microstructural damages in the cortical subcortical group, including reduced fiber density and altered diffusivity metrics. These structural abnormalities in the white matter pathways closely match the functional suppression and behavioral deficits, providing a comprehensive multimodal picture of how the brain's physical wiring can limit functional capacity. In conclusion, our study demonstrates that lesion etiology is a major determinant of neuroplasticity in hemiplegic CP. We showed that late gestation cortical subcortical injuries lead to more severe structural disconnection and functional cortical suppression compared to earlier preventricular lesions. These findings highlight the importance of etiology-specific assessments in clinical practice. By understanding these distinct patterns of reorganization, we may be able to better predict individual recovery trajectories and move toward more personalized neurorehabilitation approaches for children with early brain injuries. Finally, I would like to thank my colleagues at the Neuroscience Research Center at Cook Children's and the University of Texas at Arlington, and more importantly, the families and children who participated in this study. Thank you for your interest in our work.